Welcome back to the Physiology of the Gastrointestinal Tract. My name is Ayman Saig, and I teach at Tuskegee's Vet School. Um, uh, Before I start the lecture, I would like to draw your attention to the email address on the bottom of the screen, and it's aymansaig@hotmail.com. That's my personal email, and I'd really appreciate it if I can receive some comments or questions. Even if you have any concern, please let me know. I'll be more than happy to attend to your requests. During the past three lectures, what I've done is I've given a definition first for the GI tract, and we've decided that it's a tube-like structure that provides the body, including itself, with nutrients, fluids, and electrolytes um, through performing four functions: digestion, motility, absorption, and secretion. And I've mentioned to you that this is a complicated process that requires a control system. We've talked about the control system being intrinsic and extrinsic, meaning that there's a control system for these functions that's located within the wall of the gastrointestinal tract, and when we call that intrinsic, and there is a system that's located outside the gastrointestinal tract, and that's called extrinsic. Now, each of the systems have nerves and endocrine secretions. For the nerves in the intrinsic control system of the gastrointestinal functions, we have the enteric nervous system as well as the interstitial cells of Gahal, or the ICCs. And I've talked about the enteric nervous system, ENS, for whole lecture. And we've decided that consists of two plexuses, myenteric and submucosal, and then it has hundreds of millions of neurons, and the best way to understand it is to have it into categories to simplify understanding this topic. And we said it's based on the morphology of the cell bodies. There's Dogeal type 1 and Dogeal type 2, and based on the electrophysiological properties of the cell bodies, and we have an S-type, synaptic, and AH type, or after hyperpolarization. The third criteria was basically based on their chemical contents, or the type of neurotransmitters that they have, and we call that chemical coding. We have two types of neurons. We have excitatory, that they release acetylcholine and substance P, and we have inhibitory, that releases vasoactive intestinal peptide, or VIP, and a nitric oxide. Now, this is the neuronal part for the intrinsic control system of the gut. The neuronal part for the extrinsic innervation of the gut, or the extrinsic control systems of the gastrointestinal function, consists of two nerves, the vagus nerve and the splanchnic nerve. We've mentioned those, and we've mentioned how clinically they're relevant, and things like that. Now, in this lecture, what I will target is the endocrine secretions of the intrinsic control system of the gut, based on the roadmap slide that we talked about before. I'm going to show it to you now. Continue on the same roadmap that we've talked about. You've seen this slide before, and the highlighted ones that we've talked about already are the nerves of the intrinsic control system of the gut, which consist of the enteric nervous system and the ICCs, and the nerves of the extrinsic control system of the gut, which consists of the vagus and the sympathetic nerve, or the splanchnic nerve. Today, I will be starting the topic on endocrine secretions of the intrinsic control system of the gut. So we will be talking about the endocrine secretions of the gastrointestinal tract that participate in controlling the gut functions, the four gut functions that we've talked about. We've talked about the nerves, both inside and outside the gut. Now we will be talking about the secretions that the gut produce so it can control its own functions. And there's a couple of important facts that I would like to mention to you, and that is basically the gut is the largest endocrine organ in the body because it has millions and millions of endocrine secretory cells that secrete tons of secretions, and that is why the gut is considered the largest endocrine organ in the body because of the number of endocrine cells as well as other cells that gives secretions. The cells that participate in providing the gastrointestinal tract with secretions are mainly three types: either endocrine cells, nerve cells, and other cells. Endocrine cells, nerve cells, and other cells. I've talked about the nerves in the past 2 lectures, and I've mentioned the unique characteristic of the enteric nerve compared to other types of nerves in the body. I said these nerves, the enteric nerves, contain what we call vercosities or bulgings or swellings along the axon of the enteric nerve. These swellings are able to secrete the neurotransmitters from them, which means that the enteric nerve is able to secrete the neurotransmitter along its axon rather than from one spot, which is the presynaptic terminal in the somatic neurons. We've talked about this, and this is a very important point from a physiological standpoint because the gut is so long. It's tens of meters, so you need a neurotransmitter that is secreted along the axons so the action can happen on long distances. Now, other cells, other specialized cells, we'll be talking about them when we come to them. Now, going back, we will be talking about the endocrine cell. The endocrine cell is a pretty unique cell in itself, and I'm going to explain how the endocrine cell is a unique cell based on the shape, the structure, the ability to give secretions and things like that. We'll take one point at a time. This is a cartoon showing the shape of the endocrine cell. You will see that the endocrine cell is a triangular shape. The head of the triangle is pretty pointed, pretty narrow. It has microvilli attached to it. These microvilli function as sensors of the gut contents in the lumen of the gut. So these microvilli works as sensory receptors, sensors. They sense the environment of the gut, the internal environment of the gut, where the ingesta, where the food is located. So the pointed part of the endocrine cell is called the apex. This apex is pointed toward the lumen of the gastrointestinal tract. Again, they work as sensors. The opposite side of this apex is the base. As shown in this diagram, the base is pretty wide. The base of the endocrine cell is pretty wide. And there is a physiological reason behind that, and that is the secretions of this endocrine cell has to go to a wider area. So the sensing is going to be in a very narrow area. However, the secretions are going to be secreted into a wider space, a wider area. Narrow apex, wide base. In addition to that, you see tons of secretory granules. If you look inside the cell, you can see all of these empty circles, the purple empty circles. They are labeled secretory granules. That is because the endocrine cell is a very active cell, very active in the sense that it produces lots of secretions, So it's a very active cell. It has a mitochondria. It has an endoplasmic reticulum to generate proteins and things like that, and make energy for these secretions to take place. So these are some of the most important characteristics of the endocrine cell. Narrow apex, wide base, lots of secretory granules. A very active cell that contains mitochondria, endoplasmic reticulum, and Golgi apparatuses and other granules, which means that again it's a very active cell because it produces a lot of secretions, very active at all times. So if it's very active, how do these cells communicate with each other? How does the endocrine cells communicate with each other? We will see that they communicate in 4 different routes, or by 4 different methods. The first one is the endocrine method. Just like hormones, the secretion of the endocrine cell in the gut releases its secretions directly into the blood vessel. This is shown in the top left hand side cartoon. The secretions are secreted directly into the blood vessel. That means this is an endocrine route. The other method is neurocrine. Neurocrine means that the secretions from the endocrine cell affects the nerves in the gut, and you remember those nerves, myenteric and submucosal plexuses, hundreds of millions of neurons. We've talked about them in the past couple lectures. We call this method of communication neurocrine. So the endocrine secretion from the endocrine cell of the gut goes and reaches the neurons of the gut. this neuron then will affect an organ like a smooth muscle cell to contract or other things. So this is the second method. First one is directly dumping the secretion into the blood vessel. The second method is neurocrine, is going to the enteric neuron. The third method is called paracrine. Paracrine is that the secretion of the endocrine cell will go and affect a neighboring endocrine cell. A neighboring endocrine cell. This is called paracrine. Paracrine method of communication. The fourth and the last method of communication for the endocrine secretion is that the secretions of one cell get modified and then affect the exact same cell. Affect the exact same cell. That is called autocrine. Autocrine. Auto means self. So again, there are four methods by which the endocrine secretions communicate, or rather the endocrine cells of the gastrointestinal tract communicate with each other. One is by putting the secretions in the blood vessels, and that's an endocrine route. The other is affecting a neighboring nerve, enteric nerve, and that's called neurocrine. The other is affecting a neighboring cell, and that's called paracrine. And then the fourth and the last method is affecting its own self, and that is called autocrine. Endocrine, neurocrine, paracrine, and autocrine. Four different methods of communication. Now, where does these secretions come from then? These are the methods of communication. Where do they come from? Well, they come from three different sources, as I mentioned earlier. They come from endocrine cells. They come from nerves. They come from specialized cells. Endocrine cells, there is hundreds of millions of endocrine cells in the body, especially in the gastrointestinal tract, the largest endocrine organ in the body, the gut. And I will talk about these secretions when we come to them. Nerves is the second source of secretions. Enteric neurons, namely. Specialized cells. And the specialized cells, I will be talking about 5 of them to be specific. 5 specialized cells in the gut that will produce 5 hormones. We will be talking about them extensively. The endocrine cells will give, for example, amino acid derivatives, such as histamine. All of us heard about histamine. Endocrine cells secrete, for example, derivatives of amino acids. One example is histamine. The nerves secrete neurotransmitters. One example is acetylcholine. And you know any more examples. Substance C, P, VIP, epinephrine, norepinephrine, etc. Nitric oxide. Now, the specialized cells releases 5 different hormones. The specialized cells releases 5 different hormones. Gastrin, cholecystokinin, or CCK for short, secretin, gastric inhibitory peptide, or GIP, and then motilin. These 5 gastrointestinal hormones are secreted from 5 specialized cells in the gastrointestinal tract. 5 specialized cells in the gastrointestinal tract. So now, what I will do is I will tell you general information about these 5 hormones so you can easily memorize them. And then after that, I will talk about each of these hormones separately. I like always to go back to the base of our discussion. So everyone is following what I'm saying. First, we talked about the definition of the gut and the 4 different functions of the gut and the need for a control system that orchestrates or regulates these 4 functions of the gut. We said there is one intrinsic and the other is extrinsic. Each of these systems contain nerves and endocrine secretions. We talked about the nerves of the intrinsic control one, which is ENS and ICCs. We talked about the nerves of the extrinsic control system, which is vagus and sympathetic. Now we are talking about the endocrine secretions of the intrinsic control system, which are 5: gastrin, cholecystokinin, secretin, gastric inhibitory peptide, and motilin. These 5 hormones, these 5 endocrine secretions of the gastrointestinal tract participate in the control of the different functions of the gastrointestinal tract, which are digestion, motility, absorption, and secretion. So that's why we're going to be talking about these 5 hormones. You may ask, okay, well, you mentioned that there are hundreds of millions of endocrine cells which secrete hundreds of millions of secretions. And the answer to this is absolutely correct. But why did I choose only 5? I chose only 5 because there are only 5 secretions in the gastrointestinal tract that fulfill the criteria for being a gastrointestinal hormone. The other hundreds of thousands or millions of secretions do not fulfill the criteria to be called a gastrointestinal hormone. And that's why we specifically are talking about these 5 secretions. And I'll tell you a little later what is the criteria to be called a gastrointestinal hormone. So I did not choose these 5 haphazardly. These 5 were chosen because they fulfill the criteria to be called a gastrointestinal hormone. Just a little background about them. So we'll start talking about these 5 hormones now. Starling, the English physician, in 1905 discovered secretin and gastrin. Ivy and Oldberg in 1928, I think at the University of Chicago Medical School, discovered cholecystokinin. Brown, in the 60s and the 70s, he discovered gastric inhibitory peptide and motilin. So the 5 gut hormones, they were discovered pretty recently. Now, why did we call them gastrointestinal hormones? What is the criteria to be called a gastrointestinal hormone? There are tons of secretions, but there's only 5 secretions that fulfill the criteria to be called gastrointestinal hormones. Great. So what is this criteria? The gut hormone is a substance secreted by specialized cells in the gut. It has to be secreted by a specialized cell in the gut. Into the bloodstream of the gut. Into the bloodstream of the gut. So, the specialized cell secretes its product into the bloodstream of the gut in response to a stimuli. So this cell must be stimulated first. And what's a better stimuli in the gut than food? Food is present 24/7 in the gut. That's why it's the best stimuli for these specialized cells to secrete into the bloodstream their secretion. Now, after this secretion reaches the bloodstream of the gut, it reaches the liver by means of the portal vein. Where it gets altered in the liver. And then it passes the liver to go and reach the heart by the vena cava. After that, the heart pumps it back to the gut. Of course, to other organs as well, but we are concerned about the gut here. So it gets pumped back to the gut to basically produce its action. If the hormone contracts the gut, then the gut is going to be contracted when it receives that hormone. If the gut increases secretion of certain substances, then those substances will be secreted when that hormone reaches the gut. If the gut produces motility, then the gut will be moving when it receives that secretion, and so on and so forth. So, the gut hormone is a substance that is secreted from the gut, and it affects the gut. But after it gets modified in the liver and pumped back by the heart. Let's repeat this criteria again. What's a gut hormone? What is a gut hormone? A gut hormone is a substance secreted by a specialized cell, of course, from the gut, and reaches the bloodstream of the gut in response to a stimuli, which is in general food. Might be toxin, might be bacteria, might be any other thing, but the best stimuli and the most powerful one is food. After that, it reaches the liver, gets altered, goes to the heart, and gets pumped again to the gut to produce its functions. So the gut hormone is a substance secreted by a specialized cell from the gut, goes to the bloodstream of the gut, gets altered in the liver, gets pumped back by the heart to the gut again to produce its function. So it's produced by the gut and it functions on the gut. It affects the gut. So gut to gut. That's why it's called the gut hormone. So now, this is the definition of the gut hormone. And I said it must fulfill a certain criteria. Otherwise, it's not going to be called a gut hormone. What is this criteria? The criteria for a hormone to be called a gut hormone consists of 5 different points. That is how you're going to consider a hormone a gut hormone. First, it has to be stimulated by a physiological stimuli. So a pathological stimuli is not going to cut it. It's not appropriate because many pathologies will produce gut hormones. It's pathology. It's disease. That's not normal. We don't want that. We want something that's physiological. What's the physiological stimuli in the gut? Of course, food. So for that hormone to be called a gut hormone, it must be stimulated by food. That's criteria number 1. Criteria number 2 is that it's mostly dependent or on neuronal control, meaning that if you cut that nerve that controls this function, which is releasing this hormone, that means this hormone is not going to be released, or it's going to be attenuated. A good example of that is gastrin. A good example is CCK. And others. If you cut the vagus nerve, you will disturb the secretion of these hormones. So neuronal control should be added to the criteria to be called a gut hormone. The third criteria is that you should be able to isolate this gut hormone. You cannot say, okay, well, you have to believe me, it's a CCK, or you have to believe me, it's a GRP, or GIP, or motilin, or gastrin, or secretin. You cannot do that. You have to be able to isolate it from the gut, in the laboratory, outside. You have to show people that it's the same structure as the one that the body is secreting. The same shape, the same number of amino acids, the same structure, the same, everything is the same as the natural one. So you have to first isolate it. And then And then the fourth criteria, you have to synthesize it, as I just mentioned a second ago. Synthesize what you've isolated so you can convince people that, okay, well, this is a gut hormone. How is it a gut hormone? Because I gave the animal or the human food and I collected that product from this specialized cell. I didn't cut any of the nerves of the gut, no the vagus, not the splanchnic nerve, not the enteric neurons, nothing. All the nerves of the gut, both intrinsic and extrinsic, are intact. And then I've isolated this material. I have it in the test tube. And then not only this, but I also synthesize it. So I looked at the ones that I isolated from the animal or the human, and then I made something exactly like it in the laboratory, in the drug company, or in the peptide synthesis company. I made exactly the same substance, the same one as the one that I isolated from the animal or the human. And after that, the fifth criteria comes, I gave it to the experimental animal or to human subjects, and I recorded identical functions that we saw with the natural hormone. So whatever the natural hormone did, we recorded the exact same functions when we gave that synthesized material, synthesized substance. That's very important, very important. So, for example, if the natural substance decrease food intake, then the one that we've synthesized it outside in the drug company or in the laboratory, it must decrease food intake. If that natural hormone increases gastric acid secretion, then the one that we synthesized in the laboratory is also increasing gastric acid secretion, and so on and so forth. The functions of the synthesized substance must be identical to the functions that the natural substance produces. Identical, with no pathological effects, I may add. With no pathological effects. So if the two hormones, the natural one and the synthesized one, reduce food intake, but the one that we synthesized in the lab causes nausea, that means it's not a good one. That means it's not identical to the natural one. That's not a gut. Then that synthesized one is not a gut hormone. It only becomes a gut hormone when there is no pathological events going on. When there is no pathological events going on. Great. So now we defined what a gut hormone is. A substance secreted from a specialized cell in the gut into the bloodstream of the gut under the effect of a stimuli, which is food in general, goes to the liver by the hepatic portal vein, gets altered there in the liver, then goes to the heart by the vena cava, then gets pumped by the heart to the rest of the body by, of course, the aorta, and then reaches the gut to produce its functions. That's the definition of a gut hormone. It's released from the gut, it comes back to the gut. And the criteria for a substance to be called a gut hormone, it has to be stimulated with a physiological stimuli, such as food, and it has to be under a neuronal control, like the vagus on the splanchnic nerve or both. It should be isolated from the body, it should be synthesized outside the body, and that synthesized substance must perform the same functions as the natural hormone. This is the criteria for a gut hormone. Now, where are these 5 gut hormones coming from? This is a simple table that shows where are gut hormones being released from. Again, I mention this to simplify the study of these hormones. It's very simple when you organize them. It is pretty difficult when you study each of the hormones separately, because each of the hormones have so many characteristics and so many sites and so many things like that. So we have to put them in categories so we can identify them. This table provides information about the sites of gastrointestinal hormonal release. Only one of the hormones is from the antrum of the stomach, and that is gastrin. Only one gut hormone, 1 out of the 5, is secreted by the antrum of the stomach, and that is gastrin. All of the 5 gut hormones are secreted from the duodenum. All of them are secreted from the duodenum. Gastrin, CCK, secretin, gastric inhibitory peptide or GIP, and motilin. All of them are secreted from the duodenum. And none of them is secreted from the colon or the large intestine. Remember, in the very first lecture, I told you about the different parts of the gastrointestinal tract. I said there is the esophagus, stomach, small intestines, which include duodenum, jejunum, and ileum, and then large intestine, which includes cecum, colon, and rectum. None of the gastrointestinal hormones is being released from the large intestine. None. But all of them are released from the duodenum, and one of them is released or secreted from the antrum of the stomach. One is from the stomach, all from the duodenum, none is from the large intestine. The one from the stomach is gastrin. All of them are secreted from the duodenum. Gastrin, cholecystokinin or CCK, secretin, gastric inhibitory peptide or GIP, and then motilin. All of them are secreted from the duodenum. None of them is secreted from the large intestine. So, what do they do? This is again a simplified table of the different stimulants of gastrointestinal hormones, the 5 gastrointestinal hormones that we've mentioned, the stimulants, the type of food that stimulate the specialized cells to secrete the 5 different gastrointestinal hormones. Gastrin is stimulated by protein. CCK is stimulated by protein and fat. Secretin is called nature's anti-acid. Nature's anti-acid. So acid stimulates secretin secretion. And gastric inhibitory peptide, or GIP, is stimulated by all kinds of food, protein, fat, and carbohydrates. Motilin is related to motility. Motilin is related to motility. And we'll talk about motilin extensively when we talk about the motility of the gastrointestinal tract. But this is a simplified way so you can remember what types of food stimulate gastrointestinal hormones, or the 5 gastrointestinal hormones that we will be studying. Gastrin is stimulated mainly by protein, CCK by protein and fat, GIP by all food, secretin is by acid, and motilin is related to motility. And this slide summarizes what I just said. Gastrin is stimulated by protein, CCK is stimulated by fat and protein, GIP is stimulated by all of them, protein, fat, and carbohydrate, and secretin is called nature's anti-acid. So acid basically stimulate secretin secretion, and motilin is stimulated by fasting when there is no food in the gut. When there is no food in the gut. Again, this is a simplified picture so you can remember this for a long period of time. What are the functions that these hormones do? Gastrin stimulates acid secretion and gastric mucosal growth. Since it's the only one released from the stomach, and acid is released from the stomach, so gastrin stimulates acid secretion, and also it stimulates mucosal growth. CCK stimulates pancreatic secretions and gallbladder contraction. Pancreatic secretions, exocrine secretions, which is bicarb and some enzymes, as well as gallbladder contraction. And we'll talk about the functions of CCK again when we study each of the gastrointestinal hormones later. Secretin inhibits acid secretion. That's why it's called nature's anti-acid. And it stimulates bicarb, also to neutralize the acid secretion that makes the stomach acidic. so it stimulates bicarbs secretion, both from pancreas and from the gallbladder. Gastric inhibitory peptide is the opposite of gastrin. Gastrin stimulates acid. Gastric inhibitory peptide inhibits acid secretion. Also, it stimulates insulin secretion. Also, it stimulates insulin secretion. Motilin, again, it's related to motility. It's related to motility, both stomach and intestine. Both stomach and intestine. Let's see a summary of this slide. Gastrin stimulates acid secretion. The opposite of GIP, gastric inhibitory peptide, inhibits gastric acid secretion. Cholecystokinin, I will tell you later, the name cholecystokinin is contractor of the gallbladder. So it contracts the gallbladder, and also it increases pancreatic exocrine secretions. We'll talk about that when we talk about CCK. Secretin is nature's anti-acid. Do not forget this word. So it inhibits acid secretion, and motility is related to the motility patterns of the stomach and also in the intestine. These are the gastrointestinal hormones. 5 of them: gastrin, CCK, secretin, GIP, and motilin. They are secreted from 5 specialized cells in the gastrointestinal tract. They fulfill the definition of a gastrointestinal hormone, which is secreted by a specialized cell in the gut under the influence of a stimuli like food. This secretion goes into the bloodstream of the gut. It goes to the liver via the portal vein, gets altered, then goes to the heart via the vena cava. Then the heart pumps it back again to the gut by the aorta to produce the functions in the gut. And these 5 hormones, gastrin, CCK, secretin, GIP, and motilin, also fulfill a 5-point criteria, which they are secreted under a physiological stimuli, which is food, and we now know what type of food exactly secretes them. So gastrin is released especially by protein, CCK protein and fat, secretin is acid, GIP by all foods, protein, fat, and carbohydrates, and motilin, again, it has the motility. The second criteria is neuronal control, and usually it's the vagus. 3, we have to be able to isolate this from humans or animals or both, and then we have to synthesize them outside the body, and this synthesized material should produce the exact same function as the natural hormone or peptide. Next time, I will be talking about each of these peptides separately. I will be talking about gastrin separately, CCK separately, secretin separately, GIP separately, and motilin separately. Until that time, thank you so much for listening. And again, I would like to remind you that the personal email address, I have it on the very first slide of each of my lectures. If you have any questions or comments, please do not hesitate to write. I would love to hear from you. I would like to know. This is the these are the lectures that meant to be for first year veterinary and medical students. So I would like to hear any comments, or if you have any concerns, or any questions. If you have any questions, please let me know. I'll be more than happy to answer you. Thank you so much.